In this chapter 2, we're going to look at microscopy in the field of microbiology, where we are looking at such tiny things. How do we see them? To understand how we see them and use the microscope and the different types of microscopes, just a couple things about certain properties of light. When you are looking at light waves and how it interacts with different materials, what's going to happen as the light wave hits the material, it's either going to be reflected, absorbed, or transmitted. If it's reflected, what happens is that light wave bounces off the material. If it's absorbed, the light wave, that energy is captured by whatever type of material is. And depending on which of these things happen, it depends on the material you're talking about. Transmittance is where the light wave passes through the material. Your chapter, as you're reading it, goes into a lot of detail with the physics of light property and the wave energy, etc. We're not going to get into a lot of the physics, just we're going to be very, very basic, understanding a couple of the definitions of these terms and how it relates to using the microscope and how you use the microscope. So a couple of properties of light. Refraction, that's when light waves, as they're passing from one medium to another, they tend to change the direction. That's what refraction is referring to. The light wave bends. So like when light moves from air and then it goes to a different medium, meaning maybe through water, that is just one of the properties of light, is that light wave is going to change direction. Lenses usually are made out of glass. They tend to have a curved surface so that they will refract light. You're trying to focus that light to a certain area to produce an image. And so in these pictures, excuse me, it does show right here you have light coming through this material, and now it goes through this transparent material. Right here, see how that light wave, it bends. It doesn't go straight through in the same direction. It changes direction. It bends. There's an angle here. It's going to pass through this particular material, and now when it moves back here into this opaque material, it's going to bend again. And so that's all we're talking about, is that the differences of material and what happens to the light. In this particular image, it's showing, here comes the light wave, it hits this material. What happens is it may be reflected and bounce off, or if it goes through it, it's going to be refracted. But notice once again, it doesn't continue in this direction as you would expect as that straight line. Right here at the interface between the two materials, it's going to bend. And this is just showing it with lenses now, depending on the curve of the lens and how the light is coming through. It makes a difference as to how those light waves are going to bend. We use this like with your glasses. For those of you who wear glasses or contacts, the curvature, how is the lens made, depends on how that light's going to refract through and at what angle, where that image is going to be focused. There are different types of radiation. When we are talking about the microscope, usually what we work in a standard lab, we are just dealing with visible light. But there are also microwaves, X-rays, radio waves, UV light. Sometimes we use UV light for certain things in microbiology, and also infrared rays. So where we see the visible light, it is just a tiny small portion of the spectrum of electromagnetic waves. It's just this little tiny section right here that we're looking at. And as you can see within that visible light wave span, you've got different measurements of the wavelengths. You hit different colors. Magnification just means the ability of a lens to enlarge an object. Resolution, and you will hear this with microscopes sometimes, Resolution refers to the ability to see two items as two separate objects, and not as, oh, there's actually two things that they're so close together you're seeing it as one. Resolution, and a good microscope will have good resolution, meaning that even for very small little objects, you will see them as two separate objects. In terms of the history of using microscopes, and microscopy is using them. Robert Hooke, he used a compound microscope. Compound microscope just means you have two or more lenses. And he viewed cork, which is a plant product, and was able to see the individual cells. Antoine van Leeuwenhoek, as we mentioned in the last chapter, is the father of microbiology. He used a simple microscope. Simple microscope just means it has one lens. And he was able to view bacteria, some simple bacteria with it. So this is just showing, this is Antoine van Leeuwenhoek here. Here is his microscope that he used. As I said, it's a simple one, so it just had one lens in it, essentially kind of like a magnifying glass. And over here under C, this is Galileo. A lot of people think of him with his telescopes and his work done in astronomy. However, a lot of people do not realize that he also is considered one of the pioneers of microscopy. This is Robert Hooke's microscope, and this is drawing of what he saw, which is, you know, that's not bad, considering it was published in the 1600s. So not bad at all. But he could see the individual cells of the cork, which, as I said, that's a plant. There are lots of different types of microscopes. For your light microscopes, there's bright field, dark field, phase contrast, differential interference contrast, fluorescence, confocal scanning laser, two-photon. Regardless of what type of scope you're using, if it is a light microscope, the maximum total magnification that you can achieve, I don't care what brand it is, will be 1,000 times. Now, one advantage of working with a light microscope is you can work with either a living or a dead specimen, either one. It just depends on what you are working with. What are you trying to see? What are you trying to learn? That determines what kind of microscope you're going to use. So here's a kind of a typical bright field microscope up here at the top, and we will go over this in lab. Up here at the top, commonly referred to as the eyepiece. We in microbiology refer to it as the ocular lens. And then you have down here is this rotating nose piece that holds the objective lenses. Those can rotate around. And then we have the stage, which is where you place your specimen, the slide specimen on that. Underneath the stage, you've got a diaphragm, you've got a condenser that's going to help focus the light in. The light source on the microscope is going to be underneath and shine up through the sample. Here are the lenses, and you're looking up here. Over on the side, you have your coarse and your fine adjustment. Usually the coarse is the outer large, and the inner one, small one, is your fine adjustment. Some of the scopes are going to have a knob on the side, certainly to turn on the light, but also to adjust the light intensity. So you can dim it, you can brighten it. That's kind of your standard bright field compound microscope. In microbiology, we will be using oil immersion lens. You may or may not have used these before. Sometimes you use them in A&P, but oftentimes you don't. We need them for microbiology because we are looking at such small specimens. What the oil immersion is, the subjective lens is your 100x. That combined with the ocular lens, which is usually 10x, when you're using oil immersion, is going to give you a total magnification of 1,000 times. What you do with this is you put a little, you have your slide. I don't know if you can see right here is your slide with your specimen on it. You put a little drop of a special oil, it's called immersion oil. You put this drop, just one drop on your slide, And what it's going to do is it is going to help focus the light that's coming up from underneath, focus it all into the same direction, into that lens. It prevents the light from scattering, and this just increases or enhances the ability of you to see your specimen, because like I say, you're looking at 1,000 times magnification. Without the oil, you might see the sample, and oftentimes you will, but it's not going to be very clear because a lot of the light, if you look here, it's going to be scattering. So the lights, as it's coming through the glass, it's bending with that refraction, so not all of it is passing through the specimen and up into the objective lens. So bottom line is your sample is just not going to be very good. The resolution is going to decrease without the immersion oil. And so remember, resolution is being able to see two separate items as two separate items. It may, without the oil, it may just look like one item. The one thing I will say, learn this in lab, you do have to be careful with the immersion oil that you can only use it with the oil immersion lens, which is the 100x objective. You don't want to get it on any of the other objective lenses, so you have to be careful. Make sure you clean the slide, make sure you clean the lens, and in lab will show you how to do that, and I'll remind you of that. This is showing an example of some of the different types of things that you can see with the microscope. Bright field is where the background is light colored and your specimen is stained. In this case, obviously, it looks purple. It's very easy to see. You have a lot of little rods here and circles. You can see these look like some of your white blood cells with the nucleus in it, and these look like bacteria. They are bacillus-shaped in that they are rod-shaped, and they're in chains. Now over here on the right-hand side, this is what we call dark field. This is where the background now is dark, but the cell is light-colored, so it's the reverse. Once again, depending on what you are trying to see, sometimes you prefer one over the other. It just depends on what are you doing. Notice here these look like little spiral. They're called spirochetes. They are a type of bacteria. It's easier to see on a dark field. The clarity of it is a little bit better. Here's another example of a dark field. This is Treponema. Once again, it's a spirochete. So here it is under dark field. Here you have the bright field image, and then phase contrast. You have the same sample. It's not stained. You're just looking at it with no stain there. And this is where it's the exact same sample. Play around with the scope when you're in the lab to see what difference does it make if you adjust the amount of light, if you use a phase contrast on it. Once again, it just depends on what you're looking at and how is it that the images that you want to see. In a differential interference contrast, this is an example of what it would look like with, this is a fungus. You can see the individual cells along these strands that are known as the hyphae, and then up here on the very ends where you have the spores, the budding yeast spores, here the circular ones. You can very clearly tell this, even though it's not colored, you can very easily tell the difference between this. Sometimes we do what's known as fluorescence. This is done oftentimes what we call immunofluorescence, where we're looking for the presence of antibodies against certain pathogens. And so we have what we call direct immunofluorescence, where we put a fluorescent dye on the primary antibody itself. Here it is binding to an antigen, a foreign substance. So this blue swirly is indicative of like bacteria. Your antibody that you made against it is attached to it. You want to see if it's there. It can be hard to see. So put this fluorescent dye. It's going to glow. Indirect is where instead of putting the fluorescent dye on the antibody itself, this black structure, the one that looks like the upside down Y, you put the fluorescent dye on something that's going to bind to the first antibody. It's actually a second antibody. So it's called indirect because you're not putting the fluorescent dye on the black one, you're putting it on the green one, which then binds to the black one. There's different reasons why you may or may not want to do either process. When you look at it under the microscope, it's a fluorescent dye, so it will glow. It makes it a lot easier to very quickly see, and sometimes you may need to enumerate or count how many things you have there. Confocal microscopy, you're looking at different structures here. Now, in this case, you have, this is a cyanobacterium. It's forming a biofilm, like a sticky film on a surface, and you're wanting to look for it. You can see glycan is a carbon source, kind of like a sugar source there, but you can see the individual cells here and distinguish between them. So for a light microscopy, depending on what you want to see, will determine which kind of scope, which type of staining process you're going to use. Another type of microscope is electron microscope. There's actually two different types of electron microscopes. There's transmission, which is known as TEM, and scanning, which is known as SEM. The difference between them is that a transmission will produce a 2D image. You can see the internal structures of whatever specimen you're looking at. You have to make very, very thin slices to obtain that. Scanning will produce 3D images, but you're only going to see the surface. You will not see the internal structures. The maximum magnification is 100,000 times. So you can obviously magnify a lot more with the electron microscope than you can with the light microscope. The drawback, or one drawback of an electron microscope, is that the preparation process is going to kill your sample. And so you can only work with dead specimens. If you want to observe a living sample, you will have to use a light microscope. There's just no way around that. Another drawback is electron microscopes take some time to train, to learn how to use them. There's special techniques, and they're also very expensive. So, you know, that thing called money might play a role into whether or not you're able to use one. But they are cool to see. Over here, this image is from a TEM. You can see the internal structures, but you're only seeing 2D. Over here is the SEM, where you can see, once again, the surface, 3D structure. This is bacterium Staphylococcus aureus, but it's just the surface. You're not seeing internally. Once again, what is it you're trying to see? Helps you to figure out which one you're going to use. There's also what is known as scanning probe, scanning tunneling, STM, or atomic force, which is AFM. These do allow for the greatest magnification possible. It will allow you to see at the atomic and molecular level. And so over here is a STM image, and over here is the AFM image. Those are going to be really expensive to use. We don't have those in our lab. We don't even have electron microscopes in our lab. So usually to see something, you need to stain it because the cells are colorless, and without a stain, you're really not going to see anything. So to prepare the sample, you first are going to smear the sample on the slide, and then you're going to do what we call a heat fix it. All you're going to do is basically pass the sample, you smear it on a microscope slide, and then pass that slide through a flame. What does that do? It's going to kill it. It's going to help stick the specimen to the slide, and it's going to also allow the stain to penetrate through the cell wall better. Why are you staining it? So you can see it. It's going to provide contrast so you can actually see the cell. So this is showing different ways that you can heat fix. Some places will have a slide warmer that you just put the slide on that heat fixes it. Some of them, this is what's known as a micro incinerator, and you can hold the slide over that. We just have a flame in the lab. So if this was a burner and you had a flame, you just pass the slide through it. And sometimes you may use chemical fixation. Oftentimes in histology, which is dealing with tissue samples, they do a chemical fixation process where they stick it in a chemical. As far as the stains, we have different ways of classifying them. Basic stains, acidic stains, positive stain, negative stain. Basic stains contain positive ions. The cells usually are negatively charged, and so those positive ions are going to be attracted. Opposite charges attract. Bottom line is basic stain. If that stain has a basic charge to it, has what it's going to do, the cell is going to be colored. It will pick up that stain, that dye. Acidic stain is the reverse. It has negative ions, and so in this case what's happening is the background will be stained but not the cell. So a positive stain, which is using basic stains, you stain the cell. A negative stain stains the background. The cell's going to remain colorless. So over here is an example of a basic stain because the cells are stained. Over here as well. Over here is a negative stain, acidic stain, where the cell is clear or colorless, the background is stained. I often describe a negative stain, or I used to describe it as looking like a negative from an old camera using film. But I'm finding a lot of you now with digital everything, some of you have never seen an actual photographic negative from film. Some common types of stains. We have a simple stain, we have gram stain, acid-fast stain, capsule stains, endospore stains, flagella stains. You'll be doing these, or some of these, in the lab. In terms of, once again, types of stains, we have one classification, a simple stain versus differential. And the only difference is how many stains are you using. A simple stain just uses one stain or one dye. Examples are things like methylene blue, crystal violet, nigrosin. That's the only stain you add. So all the specimens, all the cells are going to look alike. So let's say you have bacteria and you do a simple stain and you use methylene blue. All of the cells are going to look blue, because that's the only color you used. What can a simple stain tell you? Well, you can determine the size of the cell. You can determine the morphology or the shape. Is it cocci? Is it bacillus? Is it spirochete? You can determine the arrangement. Now, one thing I'll let you know when we talk about the cells and viewing them under the microscope, morphology and arrangement are two different things. Morphology just refers to the shape. The arrangement is it singles? Is it in a cluster? Is it in a chain? Is it in pairs? That's what we're talking about for arrangement. So you can determine the size, the morphology, and the arrangement from a simple stain. A differential stain, you're using at least two different stains. So that means you're going to have two different colors in there. You can differentiate between things. Some examples are gram stain, endospore stain, and an acid-fast stain. So from a differential stain, you can still determine the size, the morphology, and the arrangement, but because of the color difference, you're going to be able to determine something else from it. Endospore stain, you'll be able to determine, does it have spores or not? The gram stain is by far the most important stain in microbiology. This is so fundamental for the classification of bacteria. It is so, so important. Nothing else is a stain. You must know and understand the process of it and how it works. With the gram stain, Now, with any of these things, first you're going to do the heat fixation. So you're going to take your sample, and you will do this in the lab. Trust me, this is one thing you have to do in the lab. You will take a sample of bacteria, and this is for bacteria. You will take your bacterial sample. It may be from a liquid broth, it may be off a petri dish, however you get it. You're going to smear it on a slide, and then you're going to heat fix it. Because remember, that's going to kill it. That's going to stick it to the slide, and that's going to make all of these stains more readily penetrate that cell wall. So after you've done that, the first stain you're going to add, we call it the primary stain, and it's crystal violet. You're going to flood that on the slide. You're going to let it sit there for 1 minute, and then you're going to rinse it off with water. Don't dry it, because you're just going to add something else to it. After you've rinsed it with water, then you're going to add what's known as a mordant. In this case, for the gram stain, it's going to be Graham's iodine. It's a special kind of iodine. A mordant just means it's going to bind to that crystal violet and make that more stabilized and hold it inside the cell wall. You're going to let the Graham's iodine stay on that slide for 1 minute. After 1 minute, you're going to rinse it with water. Then you're going to add what's known as the decolorizer. This is ethyl alcohol. There's different ways of adding this. The way I do is I add the alcohol and I let it sit on the slide for 30 seconds and then I rinse it off. Some people add the alcohol for 15 seconds, rinse, add alcohol again for another 15 seconds and rinse. Some people will add the alcohol until the slide runs clear. I find sometimes you add too much alcohol. So this is going to decolorize, and we will later talk about, both later in lecture and also in lab, why each of these steps is so important and what is actually happening. Now, after you've done the alcohol, the decolorization step, then you're going to add the second stain. This is known as your secondary stain, or some people call it your counter stain. This is saffron. Saffron is a red dye. You're going to flood that on the slide, leave it on 1 minute. After a minute, you're going to rinse with water. Now you very carefully blot it dry using bilbus paper, and then you will finally view it under the microscope. This is your gram stain. Like I say, you can determine whether the bacteria, we have two different categories. Is it gram positive or is it gram negative? You're also going to be able to tell the size, the arrangement, and the morphology. All of these are pieces of the puzzle of trying to identify an unknown bacteria. And why do you want to know how to identify your bacteria? Other than it's going to be part of your final in lab, from your standpoint, is most of you are going into the health career field. You have a patient who has an infection. How do you know what antibiotic to give them? Different microorganisms respond differently to different antibiotics. And so, like penicillin will not work with Staphylococcus aureus. So, if your patient has a staphylococcal infection, you can't give them penicillin. It's not going to work. It's not going to do anything. So how do you know if your patient has staphylococcus? Well, you have to run your lab tests. And one of the first basic steps in identification of bacteria is determining its gram reaction. Is it gram positive or is it gram negative? And so, excuse me, this table is just showing you that when you do this, the various steps of the gram stain. Step 1, when you added that crystal violet, both your gram positive, which is in this column here, versus over here, your gram negative. When you add that crystal violet, both your gram positive and your gram negatives are going to pick up that purple dye. And if you looked at it right then, it would look, both would look purple. They look the same. When you add the iodine, that mordant, it's going to help make that dye stronger, and so both are going to look purple still. When you add the alcohol, the gram positives are going to retain the purple dye, but the gram negatives are going to lose it. Why is there a difference? Because when we study the chapter on cells and talk about bacteria, you're going to find gram-positive and gram-negative bacteria have a major difference in their cell wall. And so the difference in the way they react on the gram stain, it all has to do with the differences in the structure of the cell wall. That's also going to play a role. The difference in the cell wall is going to play a role on why they respond differently to antibiotics. The fourth step of adding the saffron red. Your gram-positives are still purple. There's nowhere for the red dye to go, so they're going to remain purple. But your gram-negatives, because they were colorless from the previous step, you washed all the purple dye out, it can take up that red dye. So gram-positives are going to look purple. Gram-negatives are going to look red on your gram stain. Like I say, this is a fundamental step, so important in microbiology. This is a picture showing you have some gram-positive because you have these purple cells in here. Here's another example. This is definitely gram-positive. I can tell by the shape that they're spherical or cocci because of the arrangement that they are in these clusters. That is very unique to Staphylococcus. So my guess, usually you cannot tell genus and species from looking at a gram stain. It helps to narrow it down. But from this, because this clustering like this is unique to Staphylococcus, I could say this with pretty good accuracy that this is Staphylococcus. I just don't know which species, but it's definitely gram-positive because it's purple. See, it's all part of that puzzle. Acid-fast stain. Some bacteria, such as Mycobacterium, they have the species Mycobacterium tuberculum and Mycobacterium leprae, causative agents of tuberculosis and leprosy. Their cell wall is a little different. They have a lot of extra, what's known as mycolic acid, and it's very waxy, and so they don't gram stain very well at all. Some people will say the Mycobacterium gram stains as a gram positive, but they really don't take up that stain very well. So you have to do what's known as the acid-fast stain. In this case, you would heat fix your sample, then you add your primary stain, which is carbofuscin. This is a red stain. You have to heat this. This is a little more time consuming than the gram stain because you have to heat it for 3 minutes. You can't let it dry out while it's heating. And then you remove it from the heat and then you rinse off the carbofuscin. Then you have your decolorization step. It's acid alcohol. Gram stain uses ethyl alcohol. Acid-fast stain uses acid alcohol to decolorize. Then you add your counter stain or your secondary stain, which is methylene blue. Leave that on for a minute and then rinse it off, blot it dry. In this case, your acid-fast bacteria are going to stain red. Your non-acid-fast bacteria and your surrounding tissues, because oftentimes, like for tuberculosis, you may have tissue there from lungs. For leprosy, you might have some tissue there as well from this, the surrounding area where you took your sample. So the tissue is going to stain blue. Non-acid-fast bacteria will stain blue, but your acid-fast will look red. So in this picture here, this blue, like I said, that's your non-acid-fast, that's your tissue, but this red is your acid-fast. In this case, it's Mycobacterium tuberculosis. A capsule stain is a negative stain, so this means the background is going to stain, but the cells are going to remain colorless. It's going to make it a lot easier to see if a capsule is present. Not all bacteria produce a capsule, some do. This is important from a medical standpoint because oftentimes, if a bacteria has a capsule, that's a virulence factor. That's going to make it more pathogenic, more capable of causing a disease, causing issues. So you want to see that. Is it present or not? And so this using a couple different types of stains, but they're both negative. So you can see the background picks up the darker color. And what you're looking for is like a halo around. Endospore stains. Some bacteria produce endospores. Not all of them do, but some do. This can be important, once again, for identification purposes. Sometimes on a gram stain or even on a simple stain, if there are spores there, you kind of see like a swelling, or it's often referred to like club-shaped, like a rod with an open, large, one end is larger, so it looks like a club. But it's really best to do an endospore stain to really pick them up. In this case, what you do, primary stain, is going to be malachite green. So you heat fix it, you flood your slide with malachite green. You have to heat this again, kind of like the acid fast. In this case, you have to heat it for 5 minutes. Make sure it doesn't dry out. So you got to keep an eye on it. Don't forget about it. And then you're going to rinse it off after 5 minutes. And then your counter stain is saffron red. It's a red stain. And then rinse off after 1 minute and blot dry and then view it. With the endospore stain, the reason you have to heat it so much is for the spores, which are meant for survival, so they're very hardy. The heat is going to make it, the wall of the spore, more penetratable. From the stain's going to be able to penetrate it better. And so what happens is the endospores are going to stain green, and what we call the vegetative cell, the actively growing cell, is going to be red. So you can easily see there's a lot of spores in this particular sample. Flagella stain. Not all bacteria produce flagella. Some do, some don't. Flagella is going to help allow for motility, for movement of the bacteria. Now, as with so many things, it's going to help with identification. Is there flagella? Yes or no. The other thing, if flagella are present, that's going to help you with identification is, where is the location of that flagella? How many of them? Because the location and the number is going to be specific for that particular species of bacteria. So some bacteria will have flagella just on one end. Some will have it on both ends of the cell, if they're a rod. Some of them will have flagella surrounding the entire cell. So you do a flagella stain. Here you can see, in this case, that the flagella, look how many there are on each cell. There's lots of them. And in terms of location, they are completely surrounding the cell. It's not just on one end or just on two ends. It's surrounding it. And so once again, here's another puzzle piece for identification purposes. This is showing dark field again. Now, here's a bright field. Sometimes you want to add a stain so that you get that better contrast so you can see. So this is used all with the same organism, Treponema pallidum. This is, you can just see the differences between, here is a dark field, here is with a staining, it's a bright field, you've added a stain. You can see it a little bit better here than over here. And then this one I think is kind of cool. It is a scanning electron microscope, and you can definitely see the cell here. Kind of looks like a spring coming up. But what do you want to see? What information are you trying to determine? That helps determine what kind of a stain you're going to use, what kind of a microscope you have, and sometimes what do you have available? Because like I say, not everybody has an electron microscope available. Yes, this might be really cool to see, but we all don't have an electron microscope at our disposal. Fluorescent stains. Mentioned this a little bit earlier. This is used oftentimes in medical labs in terms of trying to determine if there's antibody or antigens from a foreign substance present, say, in somebody's maybe blood sample. What are the levels? Can you quantify? Most of you are going into the health career fields, and I can tell you most of you, or just about all of you, in your application packet are going to have to show what your antibody titers are for certain things. The antibody titer just means what is the concentration of the antibodies in your blood, the antibodies that you have against measles, against mumps, against German measles, against chickenpox, against all the various things that you've gotten your immunizations for. And so you can do a fluorescent stain as an easy way of trying to quantify, determine what is the concentration of it. Fluorescent stains are also used oftentimes in environmental settings. I actually knew a woman when I was working on my PhD. I spent some time at Oregon State University, and there's a woman there, Dr. Elaine Ingram, that developed a method. When I was there, I used to work, she'd work in the lab where I was working in at times. I was a visiting student there, so it's kind of a weird situation. I was given a lab to work in, and sometimes it wasn't her distinct lab, but that's where she would do some of her work. She developed a method she's a soil microbiologist. And oftentimes people were wondering, they had a soil sample and they were trying to figure out, per gram of soil, how many bacteria are in the sample? Or how many fungi are in the sample? And you can take the soil sample and you can dilute it out and you can get an idea of it. It's basically an estimate, and it's time consuming. So she developed a method where she took a certain type of fluorescent stain. She would take a sample of the soil, and she had all the quantities worked out. She would add the fluorescent dye, and then she would just look at that sample under the microscope. And now this was a long time ago. Okay, this would have been around 1990. And she had her microscope set up to her computer monitor, so she had a bigger screen for viewing. And she had everything calculated as to what field of view she looked at. She would add the fluorescent dye to her sample, put it under the microscope, and then she'd look at her monitor, and within a certain area, she could count. How many glowing spots did she see, essentially? She used two different dyes so that one of them stained, one of them stained yellowish green, the other one was, I believe, a reddish stain, and I forget which way it went. One was for bacteria and one was for fungi. So she could take a soil sample. People would send her samples from all over the world, and she could take the sample, she'd add the dye, she'd look at it, and she could calculate how many bacteria per gram of soil, how many fungi per gram of soil. Sometimes, like I say, there's ways that you can plate it out, but sometimes things don't grow well in the lab. And her method accounted for everything that was in there, even those that wouldn't grow, say, on a petri dish. So she was, we'll just say, quite popular. I learned very quickly. I only made the mistake once. If she was in there working, because it was fluorescent dyes, it was easy to see if the lab was dark, so she'd have the lights off. Do not walk in her lab and turn her lights on, because I made that mistake once, and she quickly informed me to shut those lights off. So I never did that again. I did, she was a pleasant lady, and once I was talking with her and asked her how long, because she would sit for hours at the microscope just counting these fluorescent spots. And so I asked her one time, how long can you sit there and do that? And she was probably in her, I would guess, her 40s then. And so she said, well, since she was getting older and she was noticing more like eye strain, she goes, I can't do it as long as I used to. She says, I can only sit here for 12 hours at a time. And I was like, oh, 12 hours, okay. But this is essentially what you're doing, whether it's environmental or medical aspect. You have this blue, which is in the schematic drawing, is a bacteria. You have an antibody, which is the black substance that's binding to it. Now, in this case, to try to really get good numbers, this is indirect. Instead of putting the fluorescent dye on the black antibody, you put it on an antibody against the antibody. It's like a second step in there, but it's going to increase your chances of being able to see it. And so that's just another example on how to use the fluorescent stains. Everything is to help you to see the microorganisms, because once again, what we're looking with, you can't see with the unaided eye. So these are techniques over time that have been developed for us to be able to see and sometimes differentiate between one strain or another.